So what is osmosis? Osmosis is a movement of a solvent such as water through a semi-permeable membrane into a solution of higher solute concentration that tends to equalize between both sides of the membrane. Osmosis is one of the most important processes in organisms such as animals, plants, and even us humans. For today's experiment, we're going to focus on plants. Osmosis is used by plants in order to absorb water from the soil. Why is that? Because the roots tend to have a higher solute concentration, and because of osmosis, water will tend to flow from the soil into the roots. Other than that, osmosis is also used to regulate guard cells. This is to allow gas exchange to occur. Talking about osmosis, we need to learn what's called tonicity. Tonicity is the ability of a solution to make water move in and out of the cell. We can divide tonicity into three terms or three solutions. First, we have hypertonic. Hypertonic is when a solution has a higher solute concentration than the cell. Here, water will move from inside of the cell into the solution. Next, we have hypotonic. Hypotonic is when a solution has a lower solute concentration than the cell. Here, water will move from the solution into the cell. And last but not least, we have isotonic. This is a condition when both the cell as well as the solution have the same solute concentration. Water still moves equally from both sides here. In this experiment, we can conclude that both apple pieces in glass A and glass B have changed in size, with the final measurement of the apple piece in glass A being 2.6 times 2.5 times 1.5 centimeters, and the final measurement of the apple piece in glass B being 2.4 times 2.2 times 1 centimeter. Both apple pieces are roughly cut around 2 times 2 times 2 centimeters. We can explain the change in size through osmosis, specifically tonicity. Now, glass A, we can conclude that glass A is a hypotonic solution, with the solution having a lower solute concentration than the apple itself. Hence why we can see that the apple has increased in size. In this picture, we can see that the apple has seemingly sunk right below the surface. This indicating that there has been a change in mass or in volume. Next, we have glass B. We can conclude that glass B is a hypertonic solution, with the saturated salt solution having a higher solute concentration than the apple piece. Water will flow from the apple piece into the solution. We can see here in this picture that the apple piece in glass B compared to glass A is still floating and seemingly has shrunk in size. There are a few factors that can affect osmosis. First, we have pressure, temperature, surface area, water potential, and concentration gradient. Pressure and temperature. The higher the value of both of these factors, the faster the molecules will move during osmosis. In this experiment, both the pressure and temperature are kept to a constant in both glass A and glass B. Next, we have surface area. The larger the surface area, the higher the rate of osmosis will be. In this experiment, both apple pieces in glass A and glass B are roughly cut around the same size, 2 times 2 times 2 centimeters. Next, we have water potential. The solution in glass A has a higher water potential for osmosis to occur compared to glass B. And last but not least, we have concentration gradient. Concentration gradient is basically the gradient or the factor that dictates whether or not osmosis can occur, since osmosis is basically water moving from a higher concentration to a lower concentration of a solute. Thank you for watching!